Hello, I'm Sarah Dickerson. I'm a scientist at Horizon Discovery, and I'll be presenting this poster on CRISPRa using synthetic guide RNA. Synthetic guide RNA is beneficial in many respects. For example, it allows for short-term expression, and you may also avoid cloning, which are some of the challenges associated with expressed guide RNAs. CRISPR can be repurposed from a gene editing tool to a transcriptional regulation system. A nucleus-deficient mutant of Cas9, which is known as dCas9, does not cleave the DNA, but retains the ability to bind guide RNAs and localize to the target DNA. When dCas9 is fused to a transcriptional regulator, it can activate transcription, commonly referred to as CRISPRa. Several CRISPRa systems have been developed, including SUNTAG, SAM, and VPR, which we show here. Initially, researchers used expressed single guide RNAs for CRISPRa, and in data not shown here, we found that the synthetic two-part CRISPRRNA and tracrRNA system can also be used to activate transcription with CRISPRa systems. Here we investigate the use of a 5' end modification of the CRISPRRNA and the 3' end modification of the tracrRNA in performing CRISPRa. We wanted to see if these modifications would have an effect on activation. So we compared modified CRISPRRNA tracrRNA, which is shown here in dark green, to unmodified, which is shown in light green, for 4 different genes, and then measured transcriptional activation via qPCR. Here we show transcriptional fold activation for individual CRISPRRNAs as well as the pools. Modifications allow for a dramatic increase in activation. Additionally, the pool of 4 CRISPRRNAs also greatly enhance activation. For example, there's more than a 30-fold increase in activation of Titin with modified CRISPRRNA pools compared to unmodified CRISPRRNA pool. To determine how synthetic guide RNAs compare to expressed single guide RNA and lentiviral expression, we compared these 3 methods for 4 different gene targets and report that synthetic CRISPRRNA tracrRNA result in gene activation comparable to expressed single guide RNA. Synthetic CRISPRRNA and tracrRNA can also be used with the SAM system. SAM is unique in that it uses the scaffold tracrRNA with an MS2 aptamer sequence that recruits transcriptional activators. We found that the transcriptional activation using either the SAM or VPR system for 6 genes was comparable. There was no consistent trend across all genes. So for example, transcriptional activation by VPR was higher for Titin, whereas transcriptional activation by SAM was higher for IL-1 receptor 2. While most of the work presented here involves UTOS cells stably expressing dCas9 and the respective activators, we also demonstrate the effectiveness in using synthetic guide RNAs with dCas9 VPR mRNA. Furthermore, the advantage to using pyrimycin dCas9 VPR mRNA or EGFP VPR mRNA is that it allows for enrichment. It is important to note that the basal expression level of the target gene may impact the level of activation achievable. Shown here, the bar graph represents transcriptional activation ranked highest to lowest for 23 different genes. The endogenous gene expression for each gene target, which was obtained from RNA-seq data, is shown on the right y-axis. We consistently found that genes that have very low endogenous expression levels are typically activated to a higher level. For instance, endogenous expression of IL-1 receptor 2 is very low, so we are able to achieve a dramatic increase in activation with CRISPRa. Synthetic guide RNAs are also effective for multiplexing, in which more than one gene may be activated. We demonstrate that the level of transcriptional activation achieved is similar whether activating a single gene, 2 genes, or on the far right of this graph, 3 genes simultaneously activated. Synthetic CRISPRa, CRISPRRNA, tracrRNA also enables pooled screening in an arrayed format. As shown here, using a cell line stably expressing dCas9 VPR allows for fast, effective screening. U2S cells stably expressing dCas9 VPR were transfected with the CRISPRa library consisting of 153 cytokine receptor targets, where each well contained a pool of 4 CRISPRa tracer RNAs targeting one gene. IL-6 secretion was then monitored by ELISA, and we were able to identify IL-6 regulators. And here in the bottom panel, we identify positive regulators of IL-6, which is shown in red, and negative regulators shown in blue. So the work presented here shows that synthetic guide RNA can be successfully used with CRISPRa systems like BPR and SAM. Multiple CRISPRRNAs targeting the same gene can be pooled to achieve higher transcriptional activation of the target gene. We also demonstrate that synthetic CRISPRRNA tracer RNA is ideal for simultaneous activation of multiple genes and is effective when used with dCas9 BPR mRNA. And finally, we show that synthetic guide RNA can be used for high throughput arrayed screening applications. Thank you so much for listening, and please feel free to reach out